Okay, welcome to those who've just joined us. We'll wait a few minutes here for people to join, and then we'll go ahead and get started. If you're just joining us, though, you can go ahead and select where you're from, and you can give us a rank from 1 to 10 of how confident you are with this topic. So we'll be doing DNA replication, and this will cover HL and SL content. Okay, we've got quite a few from Europe. I see Dubai. You'll also notice that some of the links are being put in the chat right now. So if you're looking for the PDF notes, which are the same as the slides that I'll be going through today, you can look at those. And you can also access the response form, which we'll be using to go through some practice questions during the webinar today. And finally, we have a feedback form that you can fill out when you get a chance. We would really appreciate that so that we can improve these sessions for future sessions. Okay, I think we'll go ahead and get started. Got a decent number of people right now, so that's great. All right, so I think we have everyone here. So before I introduce myself, I just want to remind you about our other Revision Village sessions, sorry, which are happening over the next couple of weeks. And they're happening mostly throughout April, and you can access the recordings for the ones that have already taken place on the Revision Village website. And you can continue to sign up for those in the next few weeks. So there's a few more to come for biology and for some of your other subjects. If you'd like to join us for those, they're all free and accessible, so please take advantage of those if you can. I would also encourage you to open, once again, those forms. So if you have the response form that you'd like to have open with us as we go through these, you can answer those questions and I'll be able to check your responses in real time to kind of see where there's confusion, and we can focus on some areas where I need to make corrections there. And if you would like a copy of the PDF notes, you can download those with the QR code that's on your screen as well for that. We also have, oops, sorry. Besides myself, we also have Phil Crick, who is helping out with some technical things and answering some of the questions in the Q&A. So if you do have questions during the presentation and you'd like to get those answered, you can message in the Q&A section, not in the chat. If you do have technical issues that you need to sort out, you can't access the notes or something like that, you can put those in the chat. But for the most part, if we keep things in the Q&A, then those answers will be accessible to everyone. So it'll be helpful. Let's see. All right, so hello, welcome to the study session on DNA replication. This is for HL and SL students. My name is Jesse Versluis, and I'm coming to you from Japan today. I am originally from Canada, and I've been teaching IB biology and environmental science and some sports science as well for close to 15 years, mostly throughout Asia. I was the head of DP Sciences at my previous school for a little over 10 years, and now I'm a full-time content developer and a tutor. So I also write some exam questions for biology and Revision Village, and possibly you've seen or heard my voice on some of the Revision Village videos for the solutions as well. So I'll be going through today DNA replication in some detail, including the various enzymes and the mechanisms that are involved, and also looking at PCR and electrophoresis for today. So we'll try and get through everything. We'll do our best to have lots of review questions as we go. And once again, if you do have any questions or anything you'd like clarified, put those in the Q&A and we can go ahead and get started. Okay, here we go. All right, so DNA replication is a requirement for all living things. How and why is the DNA replicated? Whenever cell division is taking place, we need DNA to be replicated for that. And so that's going to include growth, tissue repair, and also reproduction. So mitosis and meiosis both are going to be necessary for DNA replication, or I should say DNA replication is going to be necessary for both mitosis and meiosis. In both cases, the cells need a full copy of the genome, so they're going to be required for that. Successful DNA replication involves making identical copies of the complete genome, which is accomplished with a few things. We need free nucleotides, so those are the bases, and we also need a large variety of enzymes that are going to be involved in the process. Replication itself is considered semi-conservative, and by that we mean that part of the molecule is the original template strand of the DNA, and part of it is the newly synthesized strands. So you can see that in the center diagram here, where the template would be the blue and the red that's being formed on the top would be the newly synthesized DNA. And these are the free nucleotides that are available for that. When does it take place? It takes place during the S phase of interphase. So it takes place before cell division actually begins. So in the middle of interphase, really, you might also remember that there's a G1 and G2 phase. So this takes place in between that during the S phase or synthesis kind of phase. And in the third part of the diagram here, we can see the semi-conservative final result of DNA replication, which is that we've got our original molecule here, which is split into two template strands, the red on the top and the blue on the bottom. And then the new strands are the complementary versions of those. So we end up with two identical new DNA molecules that are formed from that. Once again, if you're just joining us, you can follow along with the PDF notes and the links for the other documents that you'll need, like the response form that we're going to go through and the feedback form, can be found in the chat. Okay, we'll also look at the DNA structure. This isn't necessarily part of DNA replication, but it's kind of necessary for you to understand some of the things we'll be going through today, like the base structures and the overall molecular structure of DNA. So the DNA nucleotide consists of a few parts, and probably you're well familiar with this at this point, but we'll go through this anyway. So we have the phosphate, which is going to form part of the backbone, and here's a phosphate molecule here, surrounded by oxygens. And we've got our deoxyribose sugars. Those are our little pentagonal structures here. And for DNA, it's deoxyribose. For RNA, it would be ribose. And then we also have our nitrogenous bases, and those are our four letters, A, T, C, and G. Since we're talking about DNA today, we won't be referring to uracil because that's only in RNA. So we've got A, C, T, and G for DNA only. We can see the full structure with all the atoms and things on the bottom left there, but we're going to use mostly the simplified version that you see in the upper diagram for today. We can also see that complementary base pairing is taking place between the bases. So we can see that, for instance, an A and a T, here's our A, here's our T, are bonded by hydrogen bonds in between. So you can see the dotted lines on the diagram represent the hydrogen bonds, and that occurs between the two strands of DNA. And then we have our covalent bonds, which are all the solid lines for everything else, essentially connecting all the phosphates and sugars to each other and to the bases themselves. The differences between purines and pyrimidines can also be seen here. So purines like A and G have two ring structures, whereas C and T, which are the pyrimidines, have only one. And you can see that an A can only bond with a T because it has the space for two hydrogen bonds that can form, whereas a C and a G can form three. So that's the structure of complementary base pairing, and it's a requirement for DNA replication because we wouldn't want an A to pair with a G or a C with a T or something like that. Because A's are always with T's and C's are always with G's, we can form two template strands that are the opposite of each other, and they can form the new DNA strand and be a perfect copy of it. Okay, so that's kind of our introduction here, and we'll have a few of these as we go through where we just check for your understanding. So this will be your first chance to try out the response form. There's no requirement for you to answer every single question or anything, but it does help us to see where there's issues. And before we go on to any questions, which we'll do in just a minute here, if you want to give the response form a try for a second and just let us know. Are you happy with the results for DNA replication? Um, yes, no. So 1, you're good. 2, so-so. 3, need some work. So I'll just check out the responses here. And then once we've got a couple answers and we're good, then we can move on. If I see too many no's, then I'll take a few more minutes and maybe um, look at that again. So it looks like everybody's good with this. That's a nice introductory topic. So we're going to go ahead and move on to some questions. And we have a mixture of both paper 1 and paper 2 questions in here. So we'll give these a try. Explain how the base sequence of DNA is conserved during replication. So this would be roughly 4-mark question. And you can go ahead and try that as well on the response form. And if you just showed up late, you can find that link for the response form in the chat. So we'll take a few minutes. You can either answer in your head, answer it on the response form, whatever you prefer. And then we'll look at the answers in a moment. Okay, we've got a couple of responses here. I'll just read one of them. So we've got adenine and guanine are 2 rings, and thymine and cytosine are pyrimidines with only 1 ring, and the DNA is semi-conservative with 1 blue original strand and another newly synthesized strand. Okay. Um, it's a decent answer. We've got some explanations that explain how it's complementary. That's good. But we can't call the strands blue and red strands because we don't know, we can't really refer to them as a color. But let's go ahead and look at the possible answers here. Okay, so DNA replication is semi-conservative, which means that we've got our template strand and our new strand. The DNA is split into 2 single strands, and then those are the template strands. Nucleotides are assembled and or attached to each single or template strand. So we've got our free nucleotides that are going to be added. They're added by complementary base pairing rules, which means that A's pair with T's and C's with G's, or you can say, give the full names, or you can say purines and pyrimidines, whatever you prefer. And then the strand that's newly formed on each template strand is identical to the original other side of the template strand. Okay, so this question specifically is referring to the semi-conservative nature of the DNA and how it's actually replicated. Not so much about the bases themselves or whatever, but just about the pairings and the semi-conservative nature there. Okay, so any 4 points from there would be your full marks for this one. Okay, here's a short answer question, just a 1-word answer. The following diagram shows the basic structure of a DNA base pair. State the names of structures X and Y. Sorry, you need 2 answers for this one, X and Y. So what's X right here and what is Y? Okay. see quite a few answers that are correct. That's great. We're getting them coming in now. All right, so X, you're probably familiar with. That would be a phosphate. So the simplified version of the phosphate can be shown as just a circle, which is nice and easy to draw. And then Y would be a hydrogen bond. So we can just say hydrogen bond in there. Okay, and then once again, here is our deoxyribose sugar. These are our bases, and then we have our hydrogen bonds in the middle, 3 of them here. Okay, great. Also note that this is pretty common for this type of question where it's worth 1 mark, but you have to have both correct answers to get that 1 mark. So if I said only phosphate and the other one was incorrect, if I said this is a covalent bond, for example, I wouldn't get any mark for this question. So these are all or nothing. There are no half marks for IB. Okay, let's move on to the next section, and then we'll have some more questions in a few more minutes here. So how do enzymes factor into the process of DNA replication? There are a lot, and actually there are more than what are even covered in the course that you're doing here. But we'll stick to the ones that are required for us. So now we're going to explore the process from start to finish. We'll skip over a couple of details that we'll come back to in terms of the leading and lagging strand, but we'll just focus just on the enzymes for the moment. So first we have helicase, and we see that here as this sort of bluish purple circle. And that's going to come in and unwind and break the hydrogen bonds that are formed between the DNA molecule, between the strands. So helicase moves along the DNA and unwinds it because it's a helix shape, obviously, and then breaks the hydrogen bonds at the same time. And this is accomplished in one direction. So it doesn't, it does it from one part of the strand, which is called the origin, and then moves towards the other end of the molecule and splits it in half. And then we get our 2 template strands that form from that. We also have DNA polymerase 3. So DNA polymerase is seen on both of the strands. Here's one, here's another. And it can be shown in many different ways. You'll see it in different colors, different sizes, different shapes, but it all represents the same enzyme. And DNA polymerase 3 is the one that actually adds the nucleotides to the template strands to form the complementary strand with base pairing rules or complementary base pairing rules. It also has the function of being able to proofread. So if there is an incorrect base, and it can actually remove it and put in the correct base instead. So that happens as it's creating the molecule. It'll stick in the nucleotides and then do a quick proofread before it moves on. And it's very accurate. It makes only a few mistakes per billion base pairs. So it's really quite an accurate enzyme at doing its job. Then we have primase, which actually isn't seen. Oh no, it is here. Here we go. Primase is only found on the lower strand in this one, although it would be on both strands in the real replication. DNA primase adds a short primer to the molecule, and that's this red section here that you can see. The primer is actually made of RNA, and so that's where DNA polymerase 3 actually attaches to the strand and begins the replication process, or begins synthesizing the new DNA. So DNA polymerase 3 can only attach to where the primer is found, and that's going to be important later on. DNA polymerase 1 is found here. It's shown as this yellow one. Its job is to remove those primers and replace them with the associated DNA. So this is pretty much the only time we're going to be talking about RNA, I think, for today. But after the strand has been synthesized, or I guess during, really, because all this is sort of happening simultaneously, DNA polymerase 1 will come in and replace those little red sections here, those primers, with the associated DNA. But then there's going to be some gaps in between where the DNA and RNA were originally like the primers and the new strand is being formed. So those little gaps need to be filled, and that's the job of the final enzyme here, which is called ligase, or DNA ligase. And it's going to join all those little disconnected fragments together to make the continuous strands that we're kind of hoping for our DNA replication. And if this all goes according to plan, then we end up with our full chromosomes and genome all duplicated, identical to the original. Okay, so we'll be talking about these enzymes a little bit more, so don't worry if you didn't quite catch all that the first time around. Okay, what are the consequences of directional DNA synthesis? So far we've only been talking about how the actual process takes place, but we need to keep in mind that the two strands, even though they're identical to each other, they're anti-parallel. So they're moving in different directions, where we have a 5' end and a 3' end for both of the strands. So the upper strand and the lower strand in this case. The origin is going to be the point where replication begins. And so what are we actually referring to with the 5' or the 3' end? That's referring to the carbons on the deoxyribose sugar. So if we were to have a sugar molecule like this, then carbon 1 would be here, 2, 3, 4, and then 5 is at the top up here, where it kind of sticks out from the rest of the molecule. But the way that DNA polymerase 3 works is it can only add bases, or sorry, it can only add new nucleotides to the 3' end, which is going to be sort of, I guess you could think of it as below, but in this case it could be either direction. So it can only add it to the 3' end of the molecule, and that's going to mean that DNA polymerase 3 can only move in one direction as it's synthesizing the DNA. So on the upper and lower template strands, it's going to move in opposite directions. So that's going to be really important in a minute here. So first we get the origin that forms, and then we get two strands as a result of this. One is called the leading strand, and the leading strand is the one that is going to follow helicase, which is the point where we get what's called the replication fork. So if we had a little helicase here, for example, it would be moving along and splitting the hydrogen bonds to expose both of the template strands. We also have primase, which is adding the primer to begin our synthesis, and then DNA polymerase will attach to those and start replicating from there. The leading strand follows the helicase on one of the strands, so DNA synthesis is always moving from 5' to 3', which means that it begins at the 5' end and adds nucleotides to the 3' end. So that's always going to be the case. And so the leading strand follows that, and it's called continuous because it's moving in the same direction. It doesn't have to take any breaks or anything. However, the other strand is going to be called the lagging strand. And in that case, the synthesis occurs away from the replication fork. So helicase is moving along the molecule in one direction, and the lagging strand is being synthesized in the opposite direction. Because of that, it's going to run into problems because you can see this on the lower diagram here. Here's helicase, and it would be moving in this direction. And on the upper strand, we can see that this would be the leading strand, which is able to continuously follow helicase and synthesize that molecule at the same time. No issues. On the bottom, as the DNA polymerase moves along, it's going to reach a point where it basically bumps into the origin where it started. And there's no exposure of new bases, and the template strand isn't available for it to make more DNA from there. So it's going to have to wait a little bit for helicase to expose more of the molecule, and then a new primer will be attached, and it can kind of leapfrog backwards and continuously make these little fragments one little section at a time. It's hard to see in a sort of two-dimensional image that isn't animated or anything, but there's some really good animations online if you ever want to go look up what that actually looks like. It can kind of help you visualize that a little bit. But because of that, it becomes discontinuous, and we have to continuously add new primers and then synthesize a little bit, wait for helicase, and then move over and do the same thing over and over again. So this is why we end up with the leading strand versus the lagging strand. And those can also be called continuous or discontinuous replication. The fragments that are formed on the lagging strand are called Okazaki fragments, after the scientist who kind of discovered those. And then finally, at the end of the process, once DNA polymerase 1 comes in and replaces the primers, then we also have DNA ligase again, which will sort of glue all the DNA together and make it a continuous strand. Okay, and we can see that again here in a larger image. So we can pay in particular close attention to the directions that we see here. So for example, our top strand is going from 5' to 3', and so we know that the strand can only be manufactured in the 5' to 3' direction. However, we've got helicase here. This is showing helicase as this structure right there that's moving along there. And we can see that our strand that's being added is in the 5' to 3' direction. So we can see the arrows are also moving in that direction, which is what we would expect. DNA polymerase 3 is at the front of that, and this is part of the leading strand here. There's 2 helicases actually in both directions. We can see one that's moving here and splitting the molecule from the origin, which would be the central part right there. One of them is moving in this direction, and the other is moving in this direction over here. So we've actually got like 2 replication forks that are moving away from each other. And we'll just focus on half the diagram at a time because it makes it a little bit confusing if we try and do the whole thing at once. So if we focus only on what's to the left of the origin, then we have our leading strand at the top. That's our leading strand right there. And it's following the replication fork, moving in the same direction as helicase here. And we've got our primer, which is shown as red there, which is where the replication began, where the synthesis of the DNA molecule began. And you can see at the 3' end right here, we're able to add new nucleotides. Here they are being added to the template and continuously moving along to follow the helicase. On the lower strand, that's our lagging strand, we've first had a primer added here, which has moved off in that direction, but we're going to ignore that one for now. The next primer would have been added here as helicase opened things up, and then DNA polymerase could synthesize this little fragment. So that's our first kind of Okazaki fragment. And then once it moves a little further, this next primer could be added, and that next section is synthesized. Again, from 5' to 3' there. On the opposite end, we can see the very end of our 3' over here. So I drew all kinds of things on here. So to make this even a little bit more complicated, just in case you're not confused already, the other end of the diagram, the one that's happening to the right, is kind of the same thing but inverted, because the strands are still 5' here to 3' on the top. And that means that on the opposite end, to the right here, we now have a lagging strand that's on the top and the leading strand, which is on the bottom. And that all depends on which part of the molecule is shown as the 5' or the 3' end. Because the strands are anti-parallel to each other, one is always going to be the leading and one is always going to be the lagging. But you can't assume that leading is always going to be on the top and lagging's on the bottom, or the other way around. It just depends on how the diagram is shown for that particular example. So pay very close attention to those if you're ever given a diagram without leading and lagging labels on it. You're looking for the strand that has 5' and 3', and you have to match it up to figure out which direction the synthesis is actually going to be taking place. So if I kind of erase all this, we can quickly look at that again. Okay, so if there were no labels whatsoever on this diagram, aside from these 4, the 5, 3, and then 3, 5 over there, I would know that the strands have to be replicated in the following directions. So because we can only add the new nucleotides on the 3' end, that doesn't mean that they're going to be added over here, because that's the template strand. So instead, the anti-parallel strand that's being synthesized is going to move in the opposite direction. So it's going to go 5' to 3', which means it has to go in this direction. And then the other strand, the one that's got a 3 on this end and a 5 over here, again, we can't just add nucleotides on the 3 end because that's again the template strand. The one that's complementary to that is going to be built in the 5 to 3 direction, which means it moves like that. So if you just remember, oh, it must be the 3' end, you might get it backwards. So you have to remember that the strand that's complementary to that, the newly synthesized ones, are going to be the opposite of the directions of the template strands. Hopefully that made sense. Okay, I know that was all confusing, and I hopefully explained that clearly enough, but if not, I feel for you. So are you happy with replication enzymes and directional synthesis? And are you ready for some more questions? So go ahead and answer that, and we can move on to the next section here. Yeah, we've got some maybes. Okay, if you do have any specific questions, again, you can ask those in the Q&A, and Phil Crick, who's helping us out today, will be happy to answer those questions as best she can. but we've got mostly yeses and some maybes. So we're going to go ahead and try some questions here. Also, before we get into this next round of questions, I'd just like to point out that we have a feedback form as well for today. If you have a chance and you're able to answer a couple of quick questions about the webinar that we're doing today, we would really appreciate you taking some time to give us some feedback and help us make some improvements. We do these every year, and we're happy to provide them as a way to help you review for your exams. But obviously, we can always make things a little bit better. So please help us out and do that. If you get a chance, we'll have the link again at the end as well. Okay, here's question 3. So what is the role of primers during DNA replication? Oh, I see a mistake. I accidentally put all A's as the answers. Just pretend it's A, B, C, D. It should show up okay on the response form. Okay, we'll just give you another minute to think about that. Although most people have the correct answer so far. Okay, great. So that's going to be B, which is that they serve as the starting point for DNA synthesis. Okay, so the primers are necessary for DNA polymerase 3 to attach and begin the synthesis. Okay, yeah, it's going to be the same for all these, I think, unfortunately. Sorry for that. Which of the following does not explain the formation of Okazaki fragments? Yeah, we've got a mix of answers for this one. Okay, so here's a question where there are multiple answers that are true, but they're not directly answering the question that's being asked of us. So we got to be careful here. So which of the following does not explain the formation of Okazaki fragments? So for example, DNA polymerase can only add nucleotides in the 5' to 3' direction. That's true. It can only add them in the 5' to 3' direction, but that doesn't explain why we would get Okazaki fragments. Not by itself anyway. Second one, same thing. DNA strands are anti-parallel to each other. Also true. And that's a part of the explanation, but the best answer here is going to be this last one, which is that the DNA primers must be added repeatedly on the lagging strand, not just a single time like they are on the leading strand. So that's going to end up producing a bunch of fragments that then need to be bonded together through DNA ligase. Okay, so it's going to be the last one for this. Okay, the diagram shows some of the enzymes involved in DNA replication. State the names of 2 enzymes used in replication not shown here. So we've got a couple of enzymes on this diagram. Which ones are not present? You have to name 2 again. So it's a single mark, but you have to have 2 enzymes and they both have to be correct. Also make sure you're specific. Okay, so we'll first start with the ones that are present here. So we've got helicase, which is shown as this one. And we also have this one, which is DNA polymerase 3. And then we also have this one here, which is going to be gyrase, I believe. But then the ones that aren't shown, there's a couple of possibilities here. We have DNA primase, which isn't present. The primers aren't shown on here at all, actually. DNA polymerase 1, which is the one that is going to replace the primers with DNA, and DNA ligase. All three of those are not here. So any two of those would be okay to get this mark. Okay, here's another one. The diagram shows part of a DNA molecule being replicated. Where would the DNA polymerase add the next nucleotide? So we've got A, B, C, D this time. Where would the next nucleotide be able to add for DNA replication that's taking place here? Okay, we got quite a few answers so far. Most of you have figured out that it should be at C here. So again, we can see we've got a 5' and a 3' end, and then on the opposite side, it's anti-parallel. We've got 3 and 5. We know that, well, hopefully we know that DNA polymerase can only add the nucleotides at the 3' end. So it could be C or B, but we also have this label on the right, and we can see that this is the template strand. So we're not adding nucleotides to the template strand because it's already finished. We need to add them to the newly synthesized strand, the one that's being built, the complementary strand. And that's going to be adding them at C. So the answer is going to be C for that question. Okay, so we'll move on to the next section here, which is about PCR and electrophoresis. Okay, so hopefully you've got the basics down for DNA replication. Now we're going to talk about a different form of replication, which is kind of artificial here. So PCR stands for polymerase chain reaction. You can see that up here. And it was developed in the 1980s. And it's really one of the most common techniques that's used for genetic engineering and for genetic testing and things like that. It's used every day in laboratories all over the world. And it's become cheap enough that even some high schools have it, and all universities really would have access to this as well. So it's used to make copies of DNA, just like real DNA synthesis in DNA replication in the cells. But this time it's not for cell division, it's for amplifying specific sections of DNA that we're interested in. And those are usually called target sequences. It could be a part of a gene, it could be like a very small section of DNA, it could be a longer section, but we're going for, instead of the full genome, just a little piece of it. And those are kind of engineered using what are called restriction enzymes. So in order to do the PCR process, we need a couple of things to make it happen. First, we need a sample of DNA. That's kind of the basis for this. And that can be obtained from lots of different things. It could be hair, it could be blood, it could be cells of some kind, tissue of some kind that we have. It could be from a crime scene, could be from a donor or something. It could be a cheek swab, if any have done the PCR COVID test, which I'm sure quite a few of you have at some point. Those are all sources of DNA that we could use. And then using the restriction enzymes, we can cut out a section of the DNA that we're actually interested in. And you can kind of see that happening here between some of those bases. Then those samples are placed into a PCR plate, and there's usually a whole bunch of little wells where they're added there. And that contains a bunch of different ingredients. Those are the DNA sample itself and some primers. Those primers are specific to be added that are a little bit different from the kind that we'll see in the cell division type. Then we've got free nucleotides. There needs to be lots and lots of nucleotides that are present to allow the DNA to be replicated. And then we've got another enzyme that we haven't seen so far, which is called Taq polymerase. It's a version of DNA polymerase 3 that is present in bacteria specifically, and they're able to function at high temperatures to allow this process to work more effectively. And then we can do this in what's called a thermal cycler, which is just a machine basically that heats up and cools down in order to artificially replicate the DNA. And we'll see how that works in a few minutes here. That happens in 3 steps. We've got first denaturation, where the DNA is split in half, and essentially the hydrogen bonds are broken. Then we've got an annealing phase where the primers are added and the replication can kind of begin from there. And that requires our polymerase enzyme. And then we've got an elongation phase where the DNA is actually synthesized and those strands are built. If that all goes according to plan, then we get our amplified DNA. So our target sequence would be copied not just once like we would get it for DNA replication. Instead, we're going to get thousands, or realistically like millions or even billions of copies of those target sequences. And the way that this works is because it's a cycler, it increases and decreases the temperature more than once. And every time it goes through a cycle of temperature changes, we get a copying of the DNA that we're after. And so if that happens a few dozen times, we end up with billions of copies of the DNA that we're interested in. So a very small sample of DNA from, let's say, a fossil or a blood sample or something like that can produce a lot of DNA. And that's going to produce better results when we actually analyze it and try and compare it to other samples and things. So that's the basics of how this works. We're going to look at the process in a little more detail in a second. Oh, hang on. I think I'm missing one. Okay, sorry about that. Yeah, I just had a computer error. Technology doesn't always cooperate. Should be able to get back into it now. All right, just give me 1 second. I'll jump back in. Okay, so picking up where we left off, we'll now look at the process of PCR, those 3 stages in a little bit of detail. So thanks again for dealing with that little technical hiccup there, and we'll hopefully pick right back up and catch up on things. Okay, so we begin with denaturation, and that is going to be where the hydrogen bonds are broken. So normally that would require helicase as an enzyme. This time we're going to do this with temperature changes. So at a high temperature, just like you'd see for a protein or something like that, the DNA will actually denature and split apart into its 2 template strands. And then we get our annealing phase where things cool down a little bit. The denaturation stage happens at a very high temperature, close to 100 degrees Celsius. Then our annealing stage happens much lower, almost half of that, maybe 45 to 65 degrees Celsius. And that is going to be where the primers actually are added. We can kind of see that. Whoops. We can see that the primers are being added. right here. And our nucleotides are starting to be added, but first we need DNA polymerase or our Taq polymerase to be useful here. So this is where our specialized enzyme is really going to come into play. So Taq polymerase is made from, or extracted, I guess, from a type of bacteria that is found in high temperatures, like hot springs, I think. And it's able to function at a high temperature without denaturing. It's an enzyme that can use those high temperatures to still do DNA synthesis, because for our own cells, that wouldn't be possible. Then the elongation phase happens at about 72 degrees Celsius. And so this allows the process to happen quite quickly and still to take place at these high temperatures. So that makes the denaturation possible, because we can do that at a high temperature without denaturing the enzymes at the same time. So we'll get to that. But those are the three phases, roughly the same as what would happen for DNA replication in cells, but this time we're only going to copy the target sequences. So the primers are only going to attach adjacent to those, not to the full genome. Okay, then electrophoresis is kind of the follow-up to a lot of PCR tests. It doesn't have to be, but a lot of the time it's connected to it and used for the actual analysis of the DNA that's produced from PCR. So if we have our amplified target DNA sequences, we have lots and lots of copies of those. We can take those and put them into an electrophoresis machine here. And how it works is we get our samples and we put them into these wells that are found along the edge of a gelatin. And that's usually agarose gel. And the gelatin itself is...
Jesse?
Yep.
Sorry, we're not in proper presentation mode. We can see your notes.
Sorry?
We can see your notes.
Oh, okay.
So it's not in proper presentation mode. Just thought I'd better let you know. I took you back as I will put you as host again, and then
Gotcha.
hopefully it'll be all right with that. That's always the problem when the computer messes up. Right, I'm going to make you host again.
Okay. Did that do it?
Yeah, that's brilliant. Thank you.
Okay. Great. Thanks for pointing that out. All right. So again, we've got our gel, we've got our samples in the gel, and now we're going to apply an electric current. So we've got our two electrodes here. We've got a negative and a positive. The DNA itself, the nucleic acids, are charged. And so they've got a charge that's going to move towards the positive here. And so there's a migration that takes place in those fragments that's going to move from the negative electrode to the positive. And as it does that, we're going to get a different amount of migration for the two segments, or for all the segments that are present, all the fragments that are in there. And you can see over here, a shorter strand is going to migrate a little bit further than a longer strand. So in the end, we get sort of a separation of the fragments that looks a little bit like this. And you've seen this, I'm sure, somewhere, these bands that form. And each sample will have a slightly different banding pattern, because hopefully they're all different DNA samples. Some of them may match, which means that they might have some similarities. Maybe they're close relatives. Maybe they're from the same source. In the case where we have two samples and we want to know if they're a match or something. So those samples can then be compared using the results of the electrophoresis. So by itself, PCR doesn't really tell us much of anything. It's just a way to make lots and lots of copies of the DNA. But the electrophoresis is able to show us the patterns, and we can compare those to make some conclusions about things. There can be lots of errors here, so it's not a perfect analysis, but it is a pretty useful tool. The samples show up much brighter when they're exposed to UV light, so that's usually what's used to kind of illuminate these things and see them a little bit more clearly, because the DNA itself is essentially transparent. You can't really see much of anything there without that UV. Okay, let me back up just a little bit for this one. So are we happy with this section for now? PCR and electrophoresis. Okay, yep, lots of thumbs up. That's great. Okay, let's try a couple more questions. So which of the following is not a stage in the PCR process? We've got elongation, annealing, termination, and denaturation. Okay, I think all the answers so far are correct, which is that termination is not one of those processes. Okay, so first we have denaturation, that's the first one, then the annealing stage, and then the elongation stage. Termination, you might be a little confused. Oh, we just did this, so you probably have them down. But if it'd been a while since you'd seen this or something, you might be thinking, no, termination sounds familiar. That's a part of transcription and translation in particular, where it has a termination phase at the end. So that might be where you've seen that term before. Okay, then we've got a 3-mark question. Outline the method that can be used to amplify small quantities of DNA to obtain large enough quantities for DNA profiling. So this is a short answer question. You got to write a little bit for this one. Give it a try in the response form if you want. And then we'll have the answers in a minute. Okay, we've got a couple answers where people are referring to the phases. That's helpful. So because it's only 3 marks, you don't have to explain everything about the process, but you would have to say some detail of it. I'll give just another minute for this one. Okay, all right. Everybody so far has definitely mentioned that it's PCR or polymerase chain reaction. Either one of those could be good enough to get you a mark. So the question specifically leaves that out to assume that you know what this process is, the one that you would be using to make amplified copies of a target sequence. We need a DNA sample, and that can be from anywhere. As long as there's DNA in it, it should work. And then we need to combine it with the materials necessary, things like the nucleotides, primers, the enzymes. If you mentioned Taq polymerase specifically, that's great. That would count. So that's another possible mark. And then we need to put it in our PCR machine or our thermal cycler. And you could go into the phases there. You could talk about elongation and denaturation and all that. And then finally, we would get a doubling of the DNA after each thermal cycle to eventually end up with all of our amplified quantity of DNA that we'd like. Okay, so only 3 marks out of these possible 5 are necessary there. Okay, now we have a little bit longer question, 6 marks this time. Compare and contrast between DNA replication in prokaryotes versus replication that occurs during the PCR process. Okay, note that this is a compare and contrast question. So we're looking for similarities and differences. So the replication that we've been looking at so far today is specifically for prokaryotes. So don't let that part throw you off. You can also answer a question like this in a table sort of form, and that can help you kind of organize your answer a little bit better without having to write full sentences necessarily. Okay, I'm going to go ahead and look at some of the answers for this. So we'll start with the similarities. So some of the similarities between the processes are they both rely on complementary base pairing. Obviously, you have to be able to match up the template strands with the nucleotides in order to make this work. They both make identical copies of the DNA. However, for the DNA replication, we're not replicating the full genome or the full chromosomes or anything like that. We're just replicating the target sequences. Both processes synthesize the DNA from 5' to 3', or you could say they add the nucleotides to the 3' end of the molecule. Both rely on enzymes for the polymerization or the synthesis of DNA. Different enzymes, but still they need enzymes. Both require primers for the initiation of DNA synthesis, and that's where either Taq polymerase or the DNA polymerase 3 will begin synthesis. And both are semi-conservative. It's not quite accurate to say that PCR is semi-conservative because the entire molecule isn't copied again, but it is still roughly a semi-conservative process because it relies on a template that can be added to. Okay, then we have our differences. So for these, you have to say both what happens for replication in cells versus PCR in order to get these marks. So that's why these bold terms like while or whereas are really important. So for instance, if I just said for this first one, PCR uses high temperatures to break hydrogen bonds, that by itself doesn't tell me what the other process does. So I need to have both in order to get these marks. So PCR uses high temperatures to break those bonds, whereas DNA replication requires helicase. PCR takes place in the thermal cycler or in vitro laboratory, while DNA replication takes place naturally or in vivo or in living things, you could say. PCR produces many copies of the DNA or replicates the DNA many times, while the DNA replication only occurs once per cell division, I guess. It occurs many, many times in your body. PCR copies the target DNA sequence only, while the DNA replication copies the entire genome. All right. And PCR makes use of Taq polymerase, while DNA replication uses DNA polymerase 3 for the synthesis of the DNA. And finally, PCR does not involve proofreading, while the DNA replication does. So those are the main differences, and we've had our similarities on the previous slide. So that's lots and lots of things you could say. There's about 12 different points there, and you'd only have to have about half of them to get those full marks. Okay, then we have a multiple choice question, which is looking at the results of electrophoresis. And it says, what conclusion can be made from the following analysis of DNA fragments? So there's not much background given for this question, and that's kind of intentional. So you'd have to look at the electrophoresis plate here and try and figure out what this test is being done for, and then look at the possible answers and try and figure out which one is probably correct. Yeah, I'll give you just another minute or so. Think about this one. Okay. So a couple more things about the electrophoresis. Usually there's like a marker which shows all the possible bands that could show up for a particular sequence. And then the samples that we're actually interested in are the ones that we're going to be comparing. So in this case, we've got a child and the mother, and we can assume that they're related to each other. And then we've got three men. So this is probably a paternity test. We can kind of assume that based on what information we're given here. But we don't necessarily need to know that because we can look at the possible answers and just see if they're true or not. So let's go through these. All three men are most likely related to each other. Uh, they have some bands in common, but for example, like this one here, everyone has that one in common. So that doesn't tell us much about them or anything like that. They do, for instance, have like all three of these in common, but there are lots of other sequences in here or fragments in here where they don't share much of anything. So we don't know enough about them to say that they're related or not. This could be a segment of DNA where everyone has kind of a similar sequence or whatever. We don't know much about this. So we probably won't say it's A. The mother has less than 50% of her genes in common with her child. If we look at the mother's fragments here, we see that she shares 1, 2 with the child, and they have 5 each. So that does kind of suggest that she shares less than 50% with the child. But we should know from basic reproduction and genetics and things like that that she's going to have roughly 50% in common with her child. That's just the way that inheritance works. So even though the number of bands for this particular segment of DNA doesn't necessarily reach that 50% level, overall, she's going to have 50% of her genome in common with her child. So it's not going to be that one. So it's not that, it's not that. All right, man number one cannot be the father. So if the child has segments that are not in common with the mother, so for instance, this one, we have to find it from the father because that kid didn't just get those um, fragments from nowhere. They got them from one of their parents. So it's not the mother. She doesn't have it right here. So we need to look for the dad that does have it. And so that's going to be possibly man two, who does have it. And if we look at his other ones that the mother doesn't have, here's one. He's got that. He's got this one. He's got this one. So most likely we could probably assume that man two out of these three people is going to be the father. And because the man number one doesn't have those segments, like he also doesn't have it here, there's no way he can be the father. So this one is going to be true in this case. The last one says man three is most likely the father. No, because if we look at the same data, we can see that man two is the one that shares those in common. So for example, for that same segment we looked at before, man three doesn't have it. So again, he can't be the father. So it's going to be C for this one, which is that man number one is probably not the father. It's probably going to be man number two. Okay, another multiple choice question here. Which of the following cannot be determined from DNA profiling using electrophoresis? So 3 of them could potentially be used, and 1 of them not so much. Yeah, we've got a mixed answers for this one, so that's interesting. Okay, so we'll just go through them one at a time here. So identification of an extinct species based on fossilized remains. Yeah, that's okay. We could potentially use it to identify if we have a sample that we could compare it to. Could we prove proof of a murder in a suspected homicide case? That one, it turns out that we probably cannot make that conclusion. So we'll come back to that one in a second for the reason why. Relatedness between two genetically similar species, no problem. We can compare that to see where they have similarities and differences. And we can use, just like we just saw in this example, evidence for paternity for an adopted child and their suspected biological parents. So that's fine. So what's the problem with B here, which is the murder in a suspected homicide case? the DNA profiling can tell us about connections between the samples that we have. So for example, I could say I have this blood that I got from a crime scene and I took some blood from some other person and I could match that to a particular person. And I could say, yes, this person probably was there at the crime. But it doesn't tell us what actually happened. It doesn't tell us there was a murder. It doesn't tell us that somebody assaulted someone else or anything like that. We need more evidence than that. We need video evidence. We need eyewitness testimony, whatever. But all it tells us is that there's a match between two people or some similarities between two samples. It doesn't tell us the full details. So DNA is a part of forensic testing that can be used for solving crimes, but it's not the full picture. Okay, here is a longer question, which is going to have multiple parts in it. And I think this is going to be our final question for today. So certain mutations are associated with increased risks of cancer. A study investigated the link between variations of the XRCC1 gene and the risk of leukemia in the Egyptian population by comparing patients suffering from leukemia with the healthy control population. Polymorphisms associated with the XRCC1 gene vary between CC, CT, and TT genotypes. The researchers suspected that one of these alleles may increase susceptibility to leukemia. So this is a question that incorporates some inheritance, but because we're near the end of the course, if you're in year 12 or grade 12, probably you're familiar with this anyway. So state two genetic techniques that could be used to identify which XRCC1 genotype participants possess. Okay, so what techniques could you use to figure out or try and figure out whether a person has CC, CT, or TT genotype? Yeah, we've got a couple of quick answers already. I think we're on the right track. Yeah, so the answers we're looking for are PCR plus electrophoresis. Okay, does that make sense? Hopefully, because we're going to take a sample from those people, we're going to amplify it, run it through the electrophoresis, and we should see some differences between those three genotypes on the banding patterns that are produced. Okay, here's the next section of the question. Evaluate the researcher's hypothesis that one allele may increase the risk of leukemia based on the results in the table below. So let's say we did our PCR, we've got a bunch of people, we're going to test them, and these are the results that we get for our three genotypes there. And the hypothesis is that one of those alleles, either a C or a T, is going to increase the risk of leukemia. Is that supported by the data that we see here? Whenever you get a question like this that says evaluate a hypothesis, usually you have to provide things that support it and some things that don't. For 3 marks, you would need to say both usually, and using the data itself or information from the question possibly. Yeah, I'll give you just another minute or so to think about it. Okay, so let's go ahead and look at some answers for this one. The question has been removed just so we can fit everything on here, but the table is still here. So we've got our genotype frequencies there, which you can kind of see. In the control group population, we can see that there's a higher proportion of Cs. CC is the most common. And in the patient group, we can see that CT and TT are much more common than they are for the control group. So that kind of indicates that there's a higher incidence or a percentage of leukemia in patients that have the T allele. So that does support the hypothesis that one of these alleles seems to be a higher risk. It also suggests that the T allele may be associated with the risk of leukemia. So that's kind of a two-part thing. One says that there is a higher percentage in those patients compared to the control group, and the other one says that that might actually be the reason why, identifying the T allele as the one that's actually causing it potentially. But this is just a correlation. We don't know for sure if this is the cause. There might be some other thing that's influencing this. So just because there's a correlation between those alleles and the patients who have leukemia, that doesn't imply that there's a specific cause. So we can point that out for a mark. And finally, the incidence of leukemia is actually greater in the heterozygous individuals, the ones that have both the C and the T. And because of that, they might actually be at a higher risk than either of the individual homozygous, either dominant or recessive groups. And so that suggests that there's a combination effect from those two alleles in the heterozygous individuals that's actually causing a greater risk. And it's not the individual alleles themselves, it's the combination of the two. So that doesn't really support the hypothesis either. So some combination of those for the 3 marks for that question would be good. Okay, and that brings us to the end of this particular webinar. So thank you again for joining us for today from Revision Village, and thank you for sticking with us, even with that little technical issue in the middle there. Thank you for answering the questions and following along with the response forms. And I hope that was helpful for your exam review coming up if you're in grade 12. And if you're in DP1, hopefully that was still helpful for your maybe mock exams or for upcoming assessments or things like that. So thank you again from myself and from Phil. And if you're able to, you can hopefully fill out the feedback form and give us a little bit of feedback for this session. Sorry. Thanks again. And I'll just remind you once more that we can, if you'd like, you can sign up for more of these sessions on the Revision Village website. We do have more for biology coming up in the next couple of weeks and also for some other subjects as well. And if you didn't get a chance, you can look at the Q&A and see some answers for questions that maybe some of your peers had